Welcome to Lesson 3 of Focal Area 2. In Lesson 1 of Focal Area 2, we learn about the 6 components of scientific project. In Lesson 2 of Focal Area 2, we learned how to ask good research questions and formulate testable hypotheses. In this lesson, we are going to learn about variables and how to design experiments. This is where your project really comes to life. You are going to learn how to set up experiments that is fair and valid. You are going to learn how to make sure that your results are trustworthy. By the end of this lesson, you will be able to identify independent, dependent, and control variables. You also be able to design an experiment that tests your hypothesis fairly. These skills are absolutely crucial for conducting a good scientific project. So let's get started. Let me start with a fundamental question. What is a variable? A variable is a factor that can change in an experiment. In any experiment, there are many factors that could potentially affect your results. Some of these factors you deliberately change, some you measure, some you keep the same. All of these are variables. There are 3 variables, and understanding the difference between them is absolutely crucial for designing a good scientific project. Let's start with the first type, the independent variable. This is what you change. You deliberately change it to see what effect it has. In a plant growth experiment, the independent variable might be the type of fertilizer. You might test 3 types: chemical fertilizer, organic fertilizer, and no fertilizer. You are deliberately changing the fertilizer to see how it affects plant growth. Here is a really important point. In a good experiment, you only change one independent variable. If you change multiple variables at once, you won't know which one caused the effect. For example, if you change both the fertilizer and the amount of water, and the plants grow taller, you won't know if it was the fertilizer or the water that caused the growth. That is why you only change one independent variable. This is called a fair test. You are testing one thing at a time. Now let's talk about the second type of variable, the dependent variable. This is what you measure. It is the result or outcome you are looking for. It changes in response to the independent variable. In the plant growth example, the dependent variable might be plant height or the number of leaves. You measure these to see if they can change when you change the fertilizer. Notice the relationship. You change the independent variable, you measure the dependent variable to see if it changes. The dependent variable depends on the independent variable. That is why it is called the dependent variable. Now, let's talk about the third type of variable, control variables. These are all the other factors that might affect your results. You keep these the same so they don't interfere with your experiment. In the plant growth example, control variables might include the amount of light, the amount of water, the type of soil, the size of the pot, and the temperature. Why is this so important? Let me give you an example. Imagine you are testing two types of fertilizer. You put one type of fertilizer on plants in a sunny window. You put the other type of fertilizer on plant in a dark corner. After a few weeks, the plants in the sunny window grow taller. Can you conclude that the first type of fertilizer is better? No, you can't conclude that because you change two things, the fertilizer and the light. The plant might have grown taller because of the light, not because of the fertilizer. That is why you need to keep control variables the same. If you kept the light the same for both groups, then you could conclude that any difference in plant growth was due to the fertilizer. This is what makes an experiment fair. You are only testing one thing at a time. You are keeping everything else the same. This is one of the most important principles in science, a fair test. When you design your experiment, you need to identify all the control variables and make sure you keep them the same for all groups. Let me really emphasize why controlling variables is so important. Imagine you are investigating how different fertilizers affect plant growth. You set up three groups of plants. Group A gets chemical fertilizer, lot of water, and lot of light. Group B gets organic fertilizer, moderate water, and moderate light. Group 3 gets no fertilizer, little water, and little light. After a few weeks, Group 1 plants are the tallest. Can you conclude that chemical fertilizer is the best? No. You can't conclude that because you change three things: the fertilizer, the water, and the light. The plants in Group 1 might have grown taller because of the fertilizer, or because of the water, or because of the light, or because of a combination of all three. You don't know which one caused the effect. Now imagine you set up the same three groups, but this time you keep the water and the light the same for all groups. All groups get the same amount of water. All groups get the same amount of light. The only thing that is different is the fertilizer. Now, after a few weeks, Group 1 plants are the tallest. Can you conclude that chemical fertilizer is the best? Yes. You can conclude that because the only thing that is different is the fertilizer. Any difference in plant growth must be due to the fertilizer. This is a fair test. This is a good experimental design. This is what makes your result trustworthy. When you design your experiment, you need to think carefully about all the factors that might affect your results. You need to identify which ones you are going to change. That is your independent variable. You need to identify which ones you are going to measure. that is your dependent variable, and you need to know or identify which ones you are going to keep the same, and those are your control variables. This is how you design a fair test. This is how you make sure your results are valid. Now, now that you understand variables, let's talk about designing the experiment. When you design an experiment, you need to keep several key principles in mind. First, use the scientific characteristics. Remember from focal area 1, science is empirical, objective, methodical, replicable, and verifiable. Your experiment should demonstrate these characteristics. Second, make it replicable. That means other people should be able to read your procedure and repeat your experiment exactly the way you did it. If they repeat your experiment, they should get similar results. This is one of the most important characteristics of science. Third, keep it methodical. That means you need clear step-by-step procedures. You need to document every detail. You need to follow your procedure exactly. Fourth, ensure validity. That means your experiment should actually test what you claim to test. If you are testing whether fertilizer affects plant growth, your experiment should test that. It shouldn't accidentally test whether light affects plant growth or whether water affects plant growth. Fifth, make it safe. You need to think about safety. Are there any hazards? Do you need safety equipment? Do you need to follow any safety procedures? When you design your experiment with these principles in mind, you produce reliable results. Your results are trustworthy, and other people can repeat your experiments and get similar results. This is what makes science powerful. When you design an experiment, there are 4 essential elements you need to include. First, a control group. The control group is the group that doesn't receive the treatment you are testing. It serves as a baseline for comparison. In the plant growth experiment, the control group would be plants that don't receive any fertilizer. This group shows what happens when you don't change the independent variable, and you compare the experimental groups to the control group to see if the treatment made a difference. Second, the experimental group or groups. These are the groups that receive the treatment you are testing. In the plant growth experiment, you might have one experimental group that receives chemical fertilizer and another experimental group that receives organic fertilizer. These groups show what happens when you change the independent variable. Third, multiple trials. You should conduct your experiment multiple times. This is called replication. Why? Because sometimes you get unusual results by chance. If you only do the experiment once and get an unusual result, you might think that's the real result. But if you do the experiment 3 times and get similar results each time, you can be confident that that's the real result. Multiple trials make your result more reliable. Fourth, careful measurement. You need to use appropriate tools to measure your dependent variable. If you are measuring plant height, you need a ruler. If you are measuring time, you need a stopwatch. You need to record exact values. You need to be consistent in how you measure. For example, if you are measuring plant height, you need to measure from the same point each time, maybe from the soil to the top of the plant. These four elements work together to ensure your experiment produces trustworthy data. When you plan your experiment, you need to think through all these elements. Let me walk you through them. First, identify your independent variable. Identify what you are going to change. Second, identify your dependent variable. Identify what you are going to measure. Third, identify your control variables. That is what you are going to keep the same. Fourth, decide on your control group and experimental group or groups. How many groups do you need? In the plant growth example, you might have 3 groups, one with chemical fertilizer, one with organic fertilizer, and one with no fertilizer. And fifth, decide how many trials you will do. How many times will you repeat the experiment? 3 times is usually a good number. C. Identify your measurement tools. What tools do you need to measure your dependent variable? Seventh, write out your step-by-step procedure. This is really important. Your procedure should be so detailed that someone else can read it and repeat your experiment exactly the way you did it. Let me give you an example. If you are investigating how fertilizers or how different fertilizers affect plant growth, your experiment plan might look like this. The independent variable will be the type of fertilizer. The dependent variable will be plant height. Control variables include light, water, soil type, pot size, and temperature. The groups, you have 3 groups, one with chemical fertilizer, one with organic fertilizer, and one with no fertilizer. Trials, you have 3 times each. And tools, you might need ruler or tape measure, measuring cups. Then procedure, you write your detailed steps. When you plan your experiment this carefully, you are setting yourself up for success. You know exactly what you are going to do. You know exactly what you are going to measure. You know exactly how you are going to keep things fair. This is good experimental design. Let's summarize what we have learned in this lesson. Variables are factors that can change in an experiment. There are 3 types: independent, dependent, and control variables. The independent variable is what you change. The dependent variable is what you measure. The control variables are what you keep the same. A fair test means changing only one variable at a time. If you change multiple variables, you won't know which one caused the effect. Good experimental design includes a control group that doesn't receive the treatment, experimental groups that do receive the treatment, multiple trials to confirm results, and careful measurement using appropriate tools. Now, before we move on to the next lesson, I want you to think about these reflection questions. First, why is it important to have a control group? What does it tell you? Second, what would happen if you change multiple variables at once? How would that affect your results? And third, can you design an experiment for a research question you are interested in? Try identify your independent variable, dependent variable, and control variables. In lesson 4 of focal area 2, we will learn how to conduct the experiment, collect data, analyze results, and draw conclusions. We will also look at real-world examples of scientific projects. So get ready for the next lesson.